Today it's my pleasure to welcome Kamil Ugurbil to our presentation for the March ReproNim webinar. Kamil is the director of the Center for Magnetic Resonance Research and professor of radiology and medicine and neuroscience at University of Minnesota Medical School. His primary research focus has been on the development and application of MR methods and instrumentation towards obtaining high spatiotemporal resolution and high accuracy functional and anatomic information in the human brain, and the development of ultra-high magnetic fields for human imaging for biomedical research in general. He's been a long-time collaborator and acquaintance of ours through the Human Connectome Project and the Brain Initiative, Connects projects, and all sorts of things that I'm sure we'll hear aspects of today. So without further ado, regarding a century of spin from electrons to precision fMRI, the floor is yours, Kamil.
Okay, well, thank you very much, David, for the invitation to present and also for the introduction. And, I was told that the seminar is very kind of flexible. I can talk about anything, but Julie put it more like, you can talk about anything you want to, and but if you can mention precision, it would be great. And but of course, I work really about on fMRI and related to the topic of precision. However, I thought for this particular topic, I will talk a little bit about the history of how MRI developed, and I will go into more precision, and I thought it may be interesting. And I came up with this idea, of course, only because this is the century of the spin, and I was asked to write introduction to a long paper that hopefully will come out, where in the introduction I summarized the history of the spin. And I thought I did all of that work for this paper. Maybe I can use some of this material for this talk as well, and that may be interesting to you. So let's see. Okay. Now, the critical experiment, if you talk about history of spin, is the Stern and Gerlach experiment. Those of you who are trained in physics would know this is a very, very famous experiment. It was conducted in 1922. And this was a very exciting time because, quantum physics was just coming up, and there were a lot of prediction and ideas, and it was really, especially in Europe, I mean, incredible amount of discoveries were ongoing. And Stern and Gerlach did this molecular beam experiment. They were really interested in testing Niels Bohr idea of the atomic structure. And in fact, Stern says that he didn't really believe what that hypothesis was, and he wanted to disprove it. And in this molecular beam experiment, in fact, they found something very unusual. Namely, it's an experiment where a beam of silver atoms are going through an inhomogeneous magnet, and they were expecting the silver atoms to have magnetic moment because of orbital angular momentum. They were not thinking about spin. The idea was not there. And in the classical picture, they would expect a uniform distribution of different angular momentums of the electron orbital moment, but they ended up seeing two actually points, and which was maybe one of the first demonstration of the quantum mechanical effect of discretization. But they didn't realize, in fact, this experiment, this splitting is due to electron spin, but they didn't realize that the idea did not exist. And they thought they were confirming the Bohr's idea that orbital angular momentum is quantized. And that came out in 1925. These two, at that time, graduate students in the lab of Paul Ehrenfest, a very famous physicist of the time. And they were graduate students, and they thought, well, no, this can be interpreted really because of if you assign a spin to an electron. So they actually use the word spin. So they introduced the concept of spin, hence the century of spin. We are a little bit past 2025, but you can talk about the century of spin because of these two graduate students. And they wrote a paper without Ehrenfeldt's name on it, but there was a footnote to this paper. It's in German by Ehrenfest, who says, "Oh yeah, well, I had some discussions with this guy or that guy about this idea, but these students had no idea about what I was discussing, and they came up with this idea by themselves." That was a footnote on that paper, which I thought was very interesting. And when you talk about spin, of course, Wolfgang Pauli comes into mind. Sometimes people assign him erroneously the discovery of spin, but he did not. He actually, however, predicted that the nucleus would have a magnetic moment. And the idea, however, he did not really have an idea that the nucleus may have a magnetic moment because it would contain a particle like proton, which is like an electron, has its own intrinsic spin. But he thought that within the nucleus there may be charged particles kind of having some dynamic behavior that results in the magnet. But he did propose the existence, and he later on got the Nobel Prize, and you may know him very well because of his exclusion principle, the Pauli exclusion principle. And later on, it was again Otto Stern who did another molecular beam experiment, this time much more precise. And instead of using silver atoms, he used hydrogen. Hydrogen has no unpaired electrons, they're paired. So he was hoping to measure then the proton magnetic moment. So this is the experiment that he now replaced the silver beam with a molecular hydrogen beam, and he measured this particular with this particular experiment, the proton magnetic moment. And based on these ideas at that time, the idea of nuclear magnetic resonance started emerging. And the very first experiment probably comes from this Dutch physicist, Cornelis Gorter, and he failed to detect it, actually. But he wrote his failed experiments as negative results of an attempt to detect nuclear magnetic spin. In 1936, he published this paper. I found these papers actually interesting to read, like this paper. The paper is in English, but the abstract in this case is in French. And he has another paper later where the abstract has two abstracts, one in German and one in French, and the paper is in English. But besides that, I mean, what he says here, for example, in the introduction, he says nuclear spin subjected to a strong magnetic field, HC, he calls it, may occupy a number of different orientations with respect to the field. And if an alternating magnetic field of frequency nu is applied perpendicularly to this field, absorption is to be expected. And he gives this formula. And this is an NMR experiment, right? Par excellence. So he had the idea, but he couldn't detect the effect. He published his negative results. And unfortunately, the sample he chose and the way he wanted to measure it was the wrong choice. But nevertheless, this is, it's in the records. And the first successful magnetic resonance experiment comes from Isadora Rabi, who performed these experiments in New York at Columbia University. And it's again a molecular beam experiment. Isadora Rabi had done something like a visiting scholar stay with Stern. He was very much Otto Stern, and he was very much aware of this, but he modified this experiment quite a bit. And he was able to, in Stern's experiment, you have an inhomogeneous field and the beam is supposed to spread. But what Rabi did is that he had two inhomogeneous fields. The beam is supposed to spread, and then the beam is supposed to refocus again. And in the middle of those two magnets, he put a homogeneous magnet and an RF coil. And so the beam spreads based on the spin state. And if you induce RF transition, it no longer focuses. And he was able to detect this. And this is the paper in Physical Review he published. It's one page, by the way. So he got the Nobel Prize for this one-page paper. And in this paper, if you look at it very carefully, you can see that he also says, oh, Gorter visited our lab and we discussed these ideas, and we thank him for his stimulating discussion. So Gorter failed, but he also inspired others, and Rabi succeeded. And in 1943 and 44, it was actually after the war, the World War II, Nobel Prizes were given for Otto Stern, who measured the magnetic moment of the proton, and Isoto Rabi in 1944 for the magnetic resonance method. Now, I showed you this instrument, and of course, this is it is really NMR, it's magnetic resonance, but it's far from anything we can do today. And that came with Edward Purcell working at MIT and Harvard, and Felix Bloch working at Stanford. And they actually showed the nuclear magnetic resonance phenomena in 1940 and got the Nobel Prize for it in 1952. So Edward Purcell really repeated the Gorter experiment, but he had a better sample, more appropriate. And when I say better, the problem with Gorter's experiments, the T1s were too long in the sample that he chose, and Purcell's T1 sample fortuitously were not that long, and he was able to detect it. And Felix Bloch actually worked with water, and his experiment really is much more akin to what we do today in terms of a MRI experiment. And of course, later on comes the big discovery for our field, namely imaging. And the idea that was unique here is to use magnetic field gradients to create an image. And as you know, there were many contributions to this idea, but Peter Mansfield and Paul Lutterworth got the Nobel Prize for that. And today, we all know that we can do obtain these fantastic anatomical images. But as you know, that my major interest is go beyond anatomical imaging, and namely functional imaging. And that development essentially started with the discovery of the Bold effect, and that was by Seiji Ogawa at Bell Laboratories. And I used to work in Bell Labs and also with Seiji, so I was very much aware of these works. And what he showed is that when you have a rat brain, and if you induce, for example, hypoglycemia, and you see these dark lines in the brain, and you can get rid of them, and then you can actually create them. And essentially, the dark lines are because of the deoxyhemoglobin, appearance of deoxyhemoglobin in the veins. And he called this the Bold effect. And this is not functional imaging, but he showed that you can manipulate the deoxyhemoglobin content, and that actually is a detectable effect in an MR experiment. But of course, we know that in the brain, we have neurovascular coupling. And from PET studies, we knew that there were changes in the oxygen consumption rate and blood flow rate. They were not exactly parallel and equivalent. And those PET studies predicted that deoxyhemoglobin would change if you have brain activity. And that idea was used in our lab in Minnesota with Seiji's participation to demonstrate, this is the first fMRI paper coming from our lab, to demonstrate that you can actually use this particular effect to detect functional images. And of course, you know that at the same time, an independent and a parallel study from MGH also presented the same discovery. And these papers, in fact, were submitted to PNAS within 5 days of each other, which is very incredible. But this is the data we published in our paper, and it was conducted at 4 Tesla, showing you the activity in the visual cortex when you have a subject sitting there and the lights are going on and off, as you saw over here. Now, we were very much interested at the time, after this data came up, really asking the question about spatial precision. So maybe this is a precision that is a little different than most of your people in ReproNim are concerned with. But nevertheless, it's a question about spatial precision. Namely, for example, all at that time and even today, we do a lot of fMRI experiments which are in the several millimeter spatial resolution. A millimeter spatial resolution is very rare, but we know, for example, about individual cortex from studies by, for example, David Hubel and Thorsten Wiesel, that there are these fine structure organizations in the mesoscopic scale. So these organizations, such as orientation domains, which is what is shown here, they got the Nobel Prize for these for discovering the orientation domains. These are neurons that respond to different orientation stimuli with different orientations. As you can see, for example, stimuli where they are moving, they have a different orientation and they're moving perpendicular to that orientation. This recording from a cell, this particular cell responds to this orientation, but certainly not to that orientation. And but they tried to even get an idea with electrophysiology how those cells were distributed, but they were completely wrong about it because of the fact that recording electrophysiology is not a very good mapping technique. And that particular map for these orientation domains actually came in 1991. It was optical imaging from Amiram Greenwald's laboratory, and they showed this fantastic map of orientation domains in the visual cortex around the singularities, called isocenters, and each color is a different orientation, and they are repeated around the singularity, and then the whole thing, the pattern repeats itself. A fantastic organization that gives you an exciting insight into how the brain has solved that problem. But it also demonstrates the power of imaging, and in addition, the fact that they were able to do it with intrinsic signals, which are hemoglobin based, gave us the idea. Okay, ask the question: can we do this with fMRI? And this is, in fact, asking the question of spatial precision, if you like, asking the question: is the neurovascular response precise enough to give you information at that particular scale? And is fMRI, does it have enough sensitivity to get to that? And we had some ideas about how to actually improve the fMRI based on a modeling paper we published. So our fMRI paper, or the MGH and our papers, came out in 1992, but in 1993 we published this paper showing that the Bold effect is, of course, mediated by blood vessels, and it comes either there are contributions coming from large blood vessels like these PL vessels or even intracortical veins. And these large blood vessels, the Bold effect for them increases with magnetic field. It increases linearly, but we don't like these large blood vessel contributions. They're a confound. They degrade spatial precision, if you like. We like Bold effects coming from these small blood vessels, namely capillaries and postcapillary venules. This effect is much, much weaker because of the fact that there's diffusion aversion comes into play. However, that weak effect scales quadratically with the magnetic field according to our modeling. And this modeling has been done by many people subsequently, and the results are more or less consistent with each other, saying that, okay, linear increase here and supralinear increases for the microvasculature. So because of that, we decided to push for high magnetic fields. So we were able to, we were lucky enough to partner with Magnex and went for a 7 Tesla project. And then we started talking about this in 1995, and in 1999, the very first 7 Tesla magnet in the world was installed in CMRR, as you can see in this picture. And with that particular magnet, we were able to, after a lot of development work, actually publish these papers showing that, yes, we can actually obtain ocular dominance columns and together with the ocular dominance columns, the orientation domains in the same subject as shown here. So this actually brought the fMRI to the world of what was possible with invasive optical imaging in animal systems. So here you see, for example, in the monkey, the orientation domains and the ocular dominance columns. The ocular dominance columns are identified by these dark lines. They are one eye versus another eye. And then the colors represent the orientation domains, and the white circles are the pinwheel centers. And this is fMRI. It's not as high a resolution as the optical imaging, but of course, fMRI can penetrate everywhere in the brain, and optical imaging can only do this in the surface. So one of the things that maybe if I can illustrate some of the things that came as a result of this kind of ability to map at that mesoscale is in a lot of work we have done in the auditory cortex. At the time, auditory cortex, when we were starting to work with 7 Tesla, people were trying to image the tonotopic maps at 1.5 Tesla, and they were simply not getting maps that sort of reproduced what was known in the animal world. But this is our first foray with 7 Tesla. We published the paper in 2003, first foray with 7 Tesla to a neuroscientific question. And we did show that, yes, just like in the animal world, human brain has mirror symmetric tonotopic maps as shown here. But we followed quite a bit on the auditory system afterwards. So we did first this tonotopic mapping, which was an accomplishment for that time. And subsequently, we showed that there is tonotopic mapping in the inferior colliculus. There is tonotopic mapping in medial geniculate body. And in fact, I don't want to go into detail, but there are other tuning characteristics which are mapped again at the scale at the same time with tonotopy in these structures. But finally, we came back to the primary visual cortex, primary auditory cortex again, showing that in the primary auditory cortex, in fact, there are columns, as you can see over here, and the tonotopic representation is columnar, spanning the cortical layers, and we were able to show, in fact, modulation of these columns at different layers based on attention. So today, this area of laminar resolution fMRI is a big area. There are many people working in this area. And it's a case where, of course, we have improved the spatial precision. However, I want to emphasize that so we were able to do that by using ultra-high fields, in this case 7 Tesla. Ultra-high field is defined as 7 Tesla and higher. An ultra-high field, however, is not just about high resolution for columns and layers. Because, I mean, if you can do those columns and layers, that means you have a lot of signal-to-noise ratio to do other things as well. I mean, the currency is signal-to-noise ratio. Ultra-high fields gives you SNR, and you can spend it however you want. You can use it to do high-resolution imaging, or you can use it to increase the contrast ratio, reproducibility, and precision of the fMRI data at any resolution. And of course, you are familiar. I mean, this is I just picked this as an example. That there are a lot of questions about the reproducibility of fMRI data. This is one paper that came out some time ago. What is the test-retest reliability? And of course, they are not talking about columns here. They're just talking about your regular millimeter, several millimeter standard fMRI, and by questioning its reproducibility. And in fact, you can even find these news articles that actually say something like, well, Duke University researchers say every brain activity study you've ever read is wrong. And why is that? Why do we have these things? Well, fMRI signals are extremely weak. They are weak. I mean, this is raw data I am showing you. This is a visual stimulation. In fact, visual responses are relatively strong, and imagine that you are doing some other complicated subtle paradigms. But in this, even with this relatively strong stimulus, the responses are small. For example, you could say that in this particular voxel, this is the time course you are seeing to this particular response. And you can say, okay, even with my eye, I can tell that there's a response that is consistent with this visual presentation, and over there also. But what about here? And maybe, yes, maybe, and there, possibly. But you see that the results are, the signal is very noisy. And often what happens in the fMRI world is that we then take these very complex statistical manipulations and try to get some inferences from it. And I have to say, when statistics gets extremely complicated, I am usually very uncomfortable about the end product. And I'd rather see these signals get much more robust. And that's what we have been really doing in pushing high fields and also pushing image reconstruction. So ultra high fields and related technologies, and these include gradients, RF coils, image acquisition and reconstruction, they increase the precision and reliability of individual measurements at all spatial resolutions. So it's not just about columns at all resolutions. And I can give you an example from the Human Connectome project that David mentioned. So the Human Connectome project, this was our, led by our consortium, Washington University, Oxford, and University of Minnesota consortium. And here you are looking at reproducibility of functional connectome, if you like. At 3 Tesla, we had this and about 185 subjects who were scanned at 3 Tesla. They were also scanned at 7 Tesla. So most of the time, people use the 3 Tesla database the Human Connectome project generated, but there's also a 7 Tesla database there, less used and fewer subjects, but it is there. And if you look at the reproducibility of functional connectomes, so this is cross-run connectome consistency, this is 3 Tesla, and below is 7 Tesla, and the reproducibility increases substantially. So, and these are not columnar resolution. The 3 Tesla data is 2 millimeter isotropic. 7 Tesla data is 1.6 millimeter isotropic. So it even actually has a slightly higher resolution, not mesoscopic resolution, factor of 2 in voxel size, but yet even with that factor of 2 decrease in voxel size, the reproducibility is substantially higher for the 7 Tesla. Now we are pushing to high fields. I mean, 7 Tesla has been successful, but in some ways we are not quite satisfied because of the fact that this mesoscopic scale studies at 7 Tesla are still at the edge. They are, they are low signal to noise, and they are not high enough resolution to really get a much better picture of what is happening in the mesoscopic scale. So we started the project, in 2008, actually, to get a 10.5 Tesla system. It took a long time to get this, but today we are working at 10.5 Tesla, and I can show you some results from them. And one of the first things that we tackled, of course, are very engineering-oriented studies. First of all, we had to develop RF coils, understand RF behavior at 10.5 Tesla, and ask questions about what is the signal-to-noise ratio gains that we expect. I mean, 7 Tesla, we knew we got some gains, but the SNR gains were still confusing. If you asked people, okay, how much of an SNR gain you get, there was still some confusing results at 7 Tesla. And I think that with 10.5 Tesla, now we can look at SNR over 3 Tesla, 7 Tesla, 10.5 Tesla. We are getting a much better appreciation of what kind of SNR gain do we get. And we did calculations, so-called ultimate intrinsic SNR. These are theoretical values, theoretically maximum SNR gains. The SNR gains are not uniform over the human head. It depends on, periphery versus sort of a central region. But in the center, over a central region, the head, for example, these yellow areas that you see in these images, you are expected to get about a factor of 2.5 compared to 7 Tesla SNR. That translates into a more than quadratic gain in SNR. And in the periphery, edge of the brain, you get slightly less than quadratic, and in the periphery, you get linear. But over, let's say, a large part of the brain or over the whole brain, you kind of have an average of quadratic increase in SNR, that you expect. And we spent a lot of time actually developing RF coils and asking the questions, can we capture that? And ultimately, I mean, there are 3 papers we published on that. I won't go in detail, but we built a lot of different RF coils, different technologies. And one of them, in fact, is this 128-channel coil at 10.5 Tesla with a lot of technological development, including, for example, on-coil preamplifiers, off-coil preamplifiers that we pull out of the transmitter so that they are not interacting, they're not getting disturbed by the transmitter, whereas the on-coil preamplifiers, we had to miniaturize them so that they don't interact with the transmitter, cable traps, and a unique sort of a self-decoupled transmitter and receiver array. The net result are some anatomical images like this, which we published. These are sagittal slices, and there is no deep learning, by the way, no denoising here. I want to mention that because, of course, today there's deep learning is used a lot, and then they can generate, deep learning also generates very pretty looking images, but these are without them, and these are high resolution, 0.2 and 0.2 in plane, 1 millimeter. So one of the slices here, for example, I'd like to call your attention to the fact that, the visual cortex, of course, occurs on both upper and lower banks of the primary visual cortex of the calcarine fissure. And this is this white area in the calcarine fissure, which is indicated here. And then you can see that the cortex has this line in the middle of it. This is the line of Jenneri, or stria of Jenneri. That is, sometimes you see it at 7 Tesla, sometimes not, and sometimes at 3 Tesla, but it's so clearly visible at 10.5 Tesla. It's really amazing. You always see it. And interestingly, in fact, there are really 2 lines there, and one of them tends to be dominant, and that's the stria of Jenneri. And so in these images, I don't want to commit myself to it, but we actually do see 2 lines in some regions of the cortex over here. And well, this made the cover page with this very beautiful depiction of the cerebellum, as you can see. This is the one I showed you was from an 80-channel coil. This is the 128-channel coil. Again, you can see the line of Jenneri extremely well and the beautiful depiction of the cerebellum. But an axial slice and showing these images like this almost doesn't do justice to the image. You can blow it up, and you can see these very nice fine structures in these images. Cerebellum, a different kind of acquisition. We are very interested in cerebellum. I will show you some fMRI data from it. EPI. And of course, when you go to high magnetic fields, EPI becomes very difficult. And of course, EPI is the workhorse of functional imaging. But at the high magnetic fields, you can also accelerate much faster. And so you get, EPI images that are of equal quality to 7 Tesla or even 3 Tesla in terms of distortions. Of course, in EPI images, there are signal losses and distortions. We all know that. They are intrinsic to it. But at 10.5 Tesla, the situation is not worse. Now, one of the things that we were very much interested in is, of course, what is the contrast to noise ratio for bold fMRI at 10.5 Tesla? So you can define this contrast to noise ratio as the signal change in an fMRI experiment versus this signal fluctuation. So it's this delta S over noise. And we know that in the gradient echo fMRI, we have these big veins I mentioned to you, and that delta S over S goes as linearly as I mentioned. And then for the capillaries, it's supposed to go roughly as a square. And what about then 10.5 Tesla data? When we do gradient recalled echo bold fMRI, how does it go? I mean, we are not distinguishing at the present time whether it goes. We are not doing it high resolution so that we can actually we are not just looking capillary areas. So this is the whole bold effect in the visual cortex in a big region of interest. And it turns out that 10.5 Tesla versus 7 Tesla, there is a bold effect increase that would be the average of these big vessels and small vessels, 2.1 times. So that actually is approximately B0 to the square. That's pretty amazing. And in fact, that suggests that really this capillary effect is now dominating at 10.5 Tesla. Otherwise, if the big vessels dominated, as they do, for example, at 3 Tesla, this is well known, 1.5 Tesla for sure, we cannot get better than linear increase in bold, but we are getting almost a square. Now we can look at what is the functional contrast ratio for fMRI. So we said delta S over N, we can rewrite it like this. And if you do high resolution, and this noise is thermal noise, then you end up with this particular equation. If we say we're looking in the thermal noise domain, and it is just delta S over S times the SNR of the image. And so this is the bold effect. And we just measured it at 10.5 Tesla versus 7 Tesla. It goes approximately B0 to the squared. And this is the image SNR. Now we have a lot of data. I showed you theoretical data, but I also showed you coils, and I didn't show you the data. We can spend a lot of time. It's published, but we actually can get SNR gains at 10.5 Tesla that matches theory. So from 3 Tesla all the way up to 10.5 Tesla, the SNR goes B0 to the B0 squared roughly. At the edges, B0, centrally more like B0 squared. So that means that functional contrast ratio for bold fMRI increases to the cube or approximately cube or the fourth power of the magnetic field. Now, when you calculate that for 3 Tesla versus 10.5 Tesla, you get these gains. 43 versus or 150, depending on we take the B3 or B4. I mean, these are huge gains compared to 3 Tesla, which is most of the fMRI world has done. Now, of course, you can say, well, you didn't take into account T1 effects and this effect, that effect. Fine. Let's throw away even 50% of that gain. You're still looking at a huge gain going to high magnetic fields. Some results from 10.5 Tesla. This is auditory cortex. We are back to auditory cortex. And now this is at 0.65 millimeter isotropic. And again, no denoising, no smoothing, fMRI images that would be, this is the highest resolution auditory cortex data you have right now. And visual cortex, these are 0.35 millimeter isotropic. And these are, for example, some of the time courses, single voxel time courses, and that you can see from this particular data. And we are interested with this looking at layers again. And what is really interesting is I showed you that gradient echo fMRI is supposed to be dominated by large vessels, but at this resolution at 10.5 Tesla, that is no longer the case. So essentially, we know that at 7 Tesla or lower magnetic fields, you have these large vessels, and the bold effect, of course, penetrates something like this red curve you see into the cortex. Sorry that the slides missed. I seem to be jumping around a little bit, but it penetrates into the cortex. And when you do, for example, fMRI at 7 Tesla, even with 0.8 millimeter resolution, across the layers, gradient echo fMRI, you see this effect, essentially the bold effect going towards the, going up with respect to towards the Peel vessels, the bald effect increases. They have this very characteristic ramping up of the bald effect across the layer. That's 7 Tesla. And so we are now looking at data at 10.5 Tesla. By the way, this now is just the EPI data. And from 10.5 Tesla on the same subject, we are looking at 7 Tesla fMRI. But at 10.5 Tesla, we are looking at anatomical images, and we can see the line of Generi. So we know that this is the middle of the cortex somewhere. And what does the 10.5 Tesla data look like? And now you still see a bit of a slope, but you see this bump over here. This is what we expect to see because the input in the visual cortex from the thalamus comes into the middle layers, and then we should see a middle layer effect. And normally we don't see it in bold, but you can see this with other techniques like vaso. And here's another subject. Again, you can see the middle, you can identify from 10.5 Tesla where is the middle of the cortex because of the fact that we can see the line of Generi even in the fMRI EPI images. And 10.5 Tesla, you see this bump, and you don't see that 7 Tesla. Just very quickly, we are very interested in hippocampus. These are hippocampus data from 10.5 Tesla, where activation is incredible and highly reproducible, as you can see it. These are divided, hippocampus divided into the sections. And I mentioned to you that we are interested in cerebellum. This is 0.5 millimeter isotropic EPI data, single EPI images. These are single EPI images from the functional imaging time series. And these are the kind of activation you see in the EPI fMRI in the cerebellum. And very, I have to say, okay, I am of course partial, but it's very exciting to see this kind of resolution and in the fMRI data, in both in visual cortex, hippocampus, and the cerebellum, and some single voxel time courses. Now, this is Eric Kandel and a picture of the book he wrote about the age of insight. And in this book, he says, central challenge of science in the 21st century is to understand the human mind in biological terms. And I think if you look back, we've had a century of development in science with respect to spins, where we are at a point, because of this development, where we can use spins really to ask questions and make inroads into this challenge that Eric Kandel mentions in this book. And I think fMRI, and particularly ultra high field fMRI, is poised to play a very central role in this aspect. Thank you very much. I have to, of course, I mentioned some of the people who you saw their names, but CMRI is a very big group of people with multiple interests and talents, and everybody really is very significant for all of those developments you talked about. And of course, we are a unique center where we have a magnet, not in a magnet bay, but in our courtyard. This is the very first 4 Tesla magnet we had for human studies, where we did the fMRI development in the old days. This is a bit of an old picture because now our courtyard has 2 magnets. One is the very first 4 Tesla, where we did the very first fMRI, and the other one is the very first 7 Tesla, which is now sitting next to it. And I mentioned some human connectome data, and David has mentioned it. So I want to also acknowledge my colleagues. This is this WashU, UMin, Oxford consortium. And particularly, I want to acknowledge my co-PI on this project, David Wanesen. So thank you very much for your attention. And that's the end of my talk.
Thank you very much. That was great and comprehensive. Before I open the questions to others, I'll take the prerogative of one. I'm pondering the relationship a bit between precision and reproducibility. Is there a position where the measurements one starts with have to be precise in order to even begin to think about the reproducibility or the generalizability of a measure? Or how do you sort of think about the relationship between precision and pushing the edges of precision and how to get field-wise, community-wise reproducibility over observations?
Well, I really, in some ways, thanks for asking that question because it does call for a clarification. I talked about two things in principle, and your question is, at least the way I understand it, you're asking me, are they the same thing? And I talked about spatial precision. That's one thing, and but I also talked about reproducibility, which is not necessarily spatial precision, but reproducibility does ask for precision in the sense that you really have to have better signal to noise and better or more accurate measurements of individual events. So if you are looking at individual subjects, and if that data from the individual subject is very noisy and you repeat that many times, your ability for reproducibility will be less. Or if you then make conclusions on that subject and you try to do it with other subjects, the ability to reproduce that will be less. So I talk about precision in two senses. Spatial precision is one. Of course, that's very important when you want to ask questions about the different levels, um, uh, different scales at which information is processed. The other one is just have to have a robust signal to noise so that your measurement actually is reproducible.
Yeah, no, I think that hits it right on the head that it's not either or, one needs, these precision methods to even have a chance to get at reproducibility
Right.
in a way that, yeah, so that's great. And it's great to have that illustrated in this way. So that's great. Other questions from the audience? I think you are able to unmute yourselves. And if any of them are shy, I have one more question in the interim. I presume your implication is not that we shouldn't be doing any pre-Tesla imaging anymore, or that we should all go out and buy a 10.5 Tesla scanner. Just like I assume the HBCD and ABCD protocols that do 10,000 subjects and get to these very high ends don't say that every study in schizophrenia or autism that I want to do need to be done in that context as well. Do you have sort of a general guideline about how mortals like us with a 3 Tesla scanner should embrace or understand or utilize the stuff that the folks who can do 10.5 Tesla? So how does that impact my life other than saying I should really save up my pennies and get a bigger scanner?
Again, David, a very important question. I am not saying at all that we should all give up 3 Tesla. And while I am saying that, laboratories like us should, we are interested in pushing the technology, and then we are anticipating that slowly that this advanced technology will filter into commercial production and to other laboratories, as it has been happening with 7 Tesla. More and more laboratories are getting 7 Tesla, and more as 7 Tesla becomes automated, more and more people are using for fMRI to generate data. But of course, I really stand behind, for example, the very large databases we have generated at 3 Tesla. They are extremely useful in many ways. A lot of work that comes out of them. What is important is that we really have to be cognizant of what are the limitations of our data. And so, for example, we have, if you look at the human connectome data, it's a 2 mm isotropic fMRI, and for 3 Tesla, and it's it's a quite robust data. There are a lot of things you can see in that data. But for example, in that particular data set, if you want to ask questions about connectivity, cortical to cerebellar or subcortical areas, it's not very good. It is just very noisy. And I do not subscribe to then doing, you know, statistical gymnastics to make conclusions out of it. Rather, if you are interested in those kind of conclusions, you can consider acquiring data with lower resolution to increase the sensitivity. Or, of course, I didn't talk about it, but as you know, there's denoising techniques you can apply, or you can go to 7 Tesla. But so we really have to be very cognizant of the limitations of the data that we have. And I think in the fMRI world, there has been so much enthusiasm, of course, coming after decades of not having a very good technique to study the brain. There has been so much enthusiasm. People have sometimes pushed the data too far.
Yeah, great. Other questions from the audience? JB unmuted. Is that for a question?
Sure. I was wondering, I mean, it's a true question. I mean, what do you think it will be the first clinical applications of very high field aspect? Because there's still, confined to the experimental labs at the moment and research labs. Do you have a sense of where the first, impact on clinic will be, Kamil?
So 7 Tesla is used today clinically, and it's approved for head imaging and knee imaging. It's not necessarily approved for the rest of the body, but I think it will come. But at the meantime, I mean, I've been watching the clinical. I mean, we don't do a lot of clinical work, but we look at it, we watch it very carefully. And there are interesting developments there. And one of the things that actually stands out right now clinically, I would say, is epilepsy with cortical dysplasia. And sometimes these, epilepsy. Well, in fact, as I said, I mean, we actually don't do a lot of clinical work, but we did a long time ago. Our clinical neurologist came to us and he said, oh, I've got this patient and I have 3 Tesla images and I think he has temporal lobe epilepsy, but I can't really conclude from the 3 Tesla. Can you help us? And we published this 2011 showing beautiful hippocampus images. These days you can get it at 3 Tesla too. That at that time you could see actually in patients that malformations in the hippocampus much better at 7 Tesla. But more so is this cortical, when we look at this cortical dysplasia in very small perturbations on the gray matter ends up being epileptic focus. And there is one famous case, and in fact, I met this individual. It's really a touching case of this young woman who was started developing epilepsy, high school, couldn't go to school, couldn't drive, couldn't go to university, and they couldn't find the epileptic. They couldn't decide where the epileptic loci was. And finally, she was enlisted in a clinical trial for 7 Tesla at Cleveland Clinic. And she had 3T images, and the radiologist, by looking at 3T and 7 Tesla images, was able to say, there was a particular area he identified in the cortex where he thought was the focus. He was able to, he was able to convince the neurosurgeons to act on it, and then they actually just drilled a hole in the skull and went in with a fiber optic and ablated that area, and she's been epilepsy free for 5 years. So there's an individual cases like this, and epilepsy is now emerging as one area where you need high resolution. Not always, but some cases you need high resolution, and where clinically it is proving to be have some advantages. Multiple sclerosis also, people talk about it as one area where they can make differential diagnosis as well.
Thank you. That's very helpful things.
Great. I know we are at time here. Any last burning questions? If not, I'd like to thank you once again for joining us. We know how to find you. If any more questions come up,
Okay.
we will happily email you.
Absolutely.
Yep. And again, I appreciate it. Great to talk to you, and we will be in touch.
Thank you very much for the opportunity, and very nice talking to you.